And our guest today is Dr. Michael Behe, who is Professor of Biological Sciences at Lehigh University in Pennsylvania. He received his PhD in biochemistry from the University of Pennsylvania in 1978. Dr. Behe is a senior fellow with Discovery Institute's Center for Science and Culture, and his research involves the delineation of design and natural selection in protein structures. In addition to publishing over 35 articles in refereed biochemical journals, he has also written editorial features in Boston Review, American Spectator, and the New York Times. His book, Darwin's Black Box, discusses the implications for neo-Darwinism of what he calls irreducible complexity, the irreducible complexity of biochemical systems. The book was internationally reviewed in over 100 publications and was named by National Review and World Magazine as one of the 100 most important books of the 20th century. Behe continues to present and debate his work throughout major universities in North America and England, and his latest book, on which he'll speak today, is The Edge of Evolution, The Search for the Limits of Darwinism, which is just out. And if you'd please join me in welcoming Dr. Michael Behe.
Well, thanks very much, Logan. It's a pleasure to be with you here today, and I'd like to thank the Discovery Institute for hosting this event. It's nice to be here in D.C. I did a postdoc after getting my PhD at Penn. I came to the National Institutes of Health, lived in Bethesda for 4 years, and so it's always nice to come back. I get to see some old friends too. Of course, D.C. has changed a little bit since 1978. Not too much, but a little bit. I talked to Logan. He said the event went from 5:30 to 7, so I made up about a little over 100 slides. And then I got a separate email yesterday that says, well, you should talk for 30 to 45 minutes. And I said, uh-oh, I'm in trouble. So what I'm going to do is I'm going to go ahead, going to try to give you the highlights, and hopefully that'll fit the time frame. I'll try to stop at least by, say, 6:30 at the latest to leave plenty of time for questions. So let me get my PowerPoint fired up here and say, okay, so the talk is the title of the book, The Edge of Evolution, and that's about as fancy as the PowerPoint gets. I know some people can do real tricks with it, but that's the best I can do. I'm from Lehigh University, as Logan said, but I'd like to say that I'm not speaking for anybody except me, and since this is just so controversial, I like to make sure that everybody knows that I'm not speaking for Lehigh University. But I'd like to start out with a little bit of a retrospective, because the new book builds on my first book, Darwin's Black Box, which was published over 10 years ago. And briefly, in that book, it was inspired by this passage in Darwin's On the Origin of Species, where he said he thought he had a good explanation for features of animals. Nonetheless, if it could be demonstrated that any complex organ existed which could not have been formed by numerous successive slight modifications, that his theory would break down. And in this, he was emphasizing that his idea of evolution had to be gradual. Random changes had to be small changes. Natural selection had to work over long periods of time. He knew that if things changed in great leaps or rapidly, then it would look like something other than random changes were involved. And in Darwin's Black Box, I said, well, one sort of system that would be a big problem for Darwin's theory is one that is irreducibly complex. And that's a fancy phrase, but it has a simple meaning. It just means that you've got some machine or system or feature of an organism that has a number of different parts, and all the parts work with each other to perform some function that none of the parts by itself could perform. And as kind of a homely example from our everyday world, I use the example of a mousetrap. A mousetrap has a number of different parts. It's got a spring, a wooden base, something called a hammer which squashes the mouse, and various other parts. And if you take any of those parts away, then the trap doesn't work. It's not like it works half as well as it used to or a quarter as well. It doesn't work at all. You've got a broken mousetrap. Now, the relevance to Darwin's theory is that it seems very hard to produce something like this by the numerous successive slight modifications that Darwin always insisted on. If you wanted to evolve something like this, you know, by something like a Darwinian process, what would you do? Would you start with, say, the wooden platform down here and hope to catch mice inefficiently, you know, maybe trip them or something? And then add another part, maybe add this holding bar thing and hope that when they trip they'd impale themselves on the holding bar. And nah, you can't do it like that. So irreducibly complex systems are big headaches for Darwin's theory. And I went on to say that there, especially in the past 50 years, science has found at the very foundation of life, the cell, many examples of irreducibly complex machinery. And perhaps the most familiar one now, 10 years down the road since Darwin's black box, is this example of what's called the bacterial flagellum, which is literally an outboard motor that bacteria use to swim. And it's got many components that are necessary for it to function. And if it's difficult to see how one would build a mousetrap gradually, it's difficult in spades to see how you would build something like this gradually. So that takes us now to the book that just came out, The Edge of Evolution. Because when I was done Darwin's black box, I had argued that there were some things in life that were beyond Darwinian explanation. But I think, and I think it's clearly true, that there are many things for which Darwin's theory is a good explanation. There are simple things that it can explain pretty well. I mean, if it can't explain the flagellum, maybe it can explain variations in the shape of your nose or something like that. You know, relatively simple things. So if that's the case, then you can say, well, how deeply into life does design extend? Or look at the other way, how much can Darwinian or other unintelligent mechanisms explain about life? Where is the edge between chance and purpose in life? And that's what the new book, The Edge of Evolution, is concerned with. And before I address that question, I want to make some distinctions, because as many of you know, when you talk about this subject, you can get yourself confused, or the conversation could be unproductive if you're not really clear what you mean by all the terms that you're using. And in particular, Darwinism, although people talk about Darwin's theory, it's really a conglomeration of little theories. There's a number of different concepts which are put together in Darwin's theory. Some parts, since it's a conglomeration of concepts, it's possible some of them might be right, some might be wrong, some might be up in the air. So I want to point out that, first of all, Darwin's theory contains at least 3 concepts. And let's tease those apart right now. One is the idea of common descent, which is that all organisms existing today have descended by a process of birth and death from organisms that lived in the misty past. And I think that's an interesting concept. There's a lot of evidence to support it, but for the problems that I am interested in addressing, it's really kind of beside the point. Maybe I shouldn't use the word trivial, but it doesn't have any explanatory power, because common descent simply is used to say that in the past some organism had a particular feature. In the present, some organism has that same feature, so maybe the present-day organism inherited from the past one. Well, that's great, and it's interesting, but it doesn't tell you where the ancestor came from. It doesn't tell you where the feature came from. It doesn't tell you how any changes occurred from the ancestor to the modern species. Darwin's theory also contains the idea of natural selection, and again, like common descent, it's kind of interesting, but ultimately not all that important in explaining life today. Natural selection simply says that, other things being equal, the more fit organisms will tend to survive and leave more offspring than the less fit organisms. something that's stronger, better adapted, or something like that, will be around longer. Well, who can argue with that? That a creature that's better will do better than a creature that's worse. And the third important part of Darwin's theory is the idea of random change. And since we've discovered DNA, that's kind of boiled down to random mutation. And this is the part of Darwin's theory that does all the work. Everything that's important in Darwin's theory is wrapped up in the concept of random mutation. And as I will argue today, that is the weakest and most poorly supported part of his theory, and really counterindicated in most instances. So we're going to focus on the concept of random mutation. Okay, so let's go back to the topic of the book. The book is called The Edge of Evolution: The Search for the Limits of Darwinism. Why think that Darwinism has any limits? It explains some things. Why not say that it can explain everything in biology? Well, there will be two reasons that I'll state today. The first thing is that life is a whole lot different than Darwin thought when he first introduced his idea in the mid-19th century. Just to concentrate on cellular and molecular matters, I'm a biochemist, that's the sort of things I deal with. Back in Darwin's day, the cell was thought to be a very simple substance. It was thought to be maybe electrified protoplasm, kind of like a little piece of jello. Nothing really that complex at all. And as a matter of fact, it was thought to be so simple that two eminent scientists of the time, Thomas Huxley and a guy named Ernst Haeckel, examined some sea mud that was brought back by an English exploring ship. And they examined some of the mud in their microscopes, crude microscopes of the day, and they thought it looked like a cell. And so they named the cell Bathybius haeckelii, after Ernst Haeckel himself, and thought that cells and new life was so simple that it could bubble up from sea mud. So that was the state of science when Darwin first proposed his theory. But now we know a lot different. Now we know that the foundation of life is considerably more complex than they knew. For example, here's the cover of an issue of the journal Cell from the late 1990s, a special review issue on the topic of macromolecular machines. Modern science has discovered that rather than being mud or a simple glob of jello, that the cell, again the foundation of life, the very basis of life, is enormously complex. It's like a nanoscale factory filled with sophisticated machinery. And if you turn and you look at the table of contents of that issue of the journal Cell, you'll see papers with titles such as "The Cell is a Collection of Protein Machines," "Polymerases in the Replisome," "Machines Within Machines," "Mechanical Devices of the Spliceosome," "Motors, Clocks, Springs, and Things." So the point is that rather than a drop of mud, the cell is much more like a very modern factory, computer-controlled, completely automated. Here's a computer illustration of just one of those machines. I won't go into how it works or anything, but I just want to show this picture of this, which is called the eukaryotic cilium, to give you a taste of the structural and mechanical complexity of one of the machines. All of these little blue, lighter and darker blue dots, the green and white, are made of things called proteins, which are kind of the basic substance of the cell, which in themselves are complex objects, and they all have to fit together to form this really what is a motorized paddle, oftentimes used in the cell. The cilium is composed of hundreds of different protein parts, all of which have to find each other in the cell, assemble and fit together to give this working motorized paddle. And you get a feeling for how complex this is simply by where this illustration is published. It's published in the Journal of Nanotechnology. just like the technology that humans are now starting to invent to manipulate things at the atomic level. The cell has been doing it for a long, long time. So one reason we can think that Darwinism has limits is that life has turned out to be much different, much more sophisticated than was initially thought. A second reason is this. It's called the problem of the rugged evolutionary fitness landscape. And most people probably haven't heard about these things, but in the early part of the 20th century, there was a mathematician named Ronald Fisher who was interested in biological problems as well, who said, well, let's think of evolution as a hill and say that a species that's not fit so well is at a lower part of the hill and has particular DNA, and maybe by a mutation it can get higher on the hill and higher till it reaches the zenith. And that's kind of a popular view of evolution. You know, mutation comes along, the organism gets better, another one comes along, gets even better until it's great. But now that we know the complexity of life, this simple, almost cartoonish conception is really inadequate. As many people have talked about, many scientists have talked about, a more realistic evolutionary landscape would be quite rugged, where you could change one component, one nut or bolt of a molecular machine. That might help a little bit, but then to help something else, maybe you could change some other completely different component. Now, the important point about this is that in a situation like this, there's a direction to evolution. It can go up, it goes up until it gets the best it can be. But if you're an organism that's on this peak of a rugged landscape, there's no direction. You could go this way or that way or the other way. You could change a nut or a bolt or a lever or something, but you're not really accumulating something beneficial. And the landscape, such a landscape is what I call incoherent. It's substantially incoherent. That means, again, there's no particular a change in one feature is not necessarily correlated with a change in another feature that's going to help the organism. There's no net benefit in any particular single machine. And scientists have been recognizing this. There have been a number of books in the past 10 years written by people of good reputation, not like me, who challenge Darwin's theory and say it's not adequate to explain what we know about biology. One guy named Richard Watson, an Englishman, wrote a book called Compositional Evolution last year. He's a computer scientist interested in evolutionary computation. And he says, in computer science, we recognize that the algorithmic principle described by Darwin as hill climbing, more specifically random mutation hill climbing. However, we also recognize that hill climbing is the simplest possible form of optimization and is known to work well only on a limited class of problems, kind of like on antibiotic resistance and some things I'll talk about in a few minutes. So things have changed for Darwin. Life is a whole lot more complex. We recognize that his idea, although it works on some things, doesn't work on much. And so there are good reasons to think that Darwin's theory is limited. Nonetheless, a lot of scientists, as you may know, think that Darwin's theory is the explanation for all of life. For example, Richard Dawkins, who's been in the news this past year or so on more philosophical topics, wrote in 1986 that he says, "We have seen that living things are too improbable and too beautifully designed to have come into existence by chance. How then did they come into existence? By gradual, step-by-step transformations from simple beginnings." Kind of like the simple hill, the simple evolutionary landscape that I showed you. So he doesn't let these things, these little technicalities, these little complexities of life bother him. He says that, you know, no matter what, we think that Darwin's theory can explain everything. So it's kind of like this, the situation is, and this is just a cartoon I got off the internet. You know, we've got a fellow here stranded on an island, And he's thinking to himself, well, I'll just write a note in a bottle and I'll throw it out into the ocean, and certainly, you know, I'll find, you know, somebody will come to rescue me, and it will probably be somebody like that. And yet, if you sit down and calculate coldly what's the likelihood of that happening, the odds are against it. So things have boiled down, especially since Darwin's Black Box was published 10 years ago, to a group of people like intelligent design proponents and even other folks like Richard Watson saying Darwin's theory doesn't work. Other people saying, yes, it does. Other people saying, no, yes, no, yes. So it's kind of stalemated, you know, yes, you know, says so, says not, says so, says not. So what we need is evidence. And that's what the edge of evolution focuses on. It asks the question, okay, well, leave aside our imaginations, what we want. What's the evidence show that Darwinian processes do? In my book, I talk about the fact that the best evidence to assess the ability of Darwinian processes comes from studies of malaria, because malaria has plagued the human race for a long time, and in this genomic era, we have been able to track down genetic changes in the human genome, which has given people a portion of a defense against malaria. Additionally, in recent years, just the past 5 years or so, we've also been able to track down genetic changes in malaria as it developed resistance against drugs that we use to treat people. And it turns out because of our detailed genetic studies of this evolutionary conflict between us and a malarial parasite, and because of something I'll talk about later, just the sheer number of parasites that infest humanity from malaria, this is the best evidence for what Darwinian processes can do. So let's start with a little overview of this. You know, I know this is a little more technical and a little more outside of what's normally discussed in evolution, so bear with me. I think the result will be rewarding. So when somebody is bitten by a malaria-infected mosquito, the malaria parasite, which is a single-celled creature, gets into your body, goes to your liver, hangs out there a little while, reproduces until it makes thousands of kind of preliminary cells, which go and invade red blood cells. See, they attach themselves to a red blood cell, they go inside, and they eat the hemoglobin. They eat the insides of the red blood cell. And once they have done that, they multiply to about 20, and they burst out of the red blood cell, and each of the 20 goes and attaches itself to a new red blood cell where the process reproduces. So they eat your blood. And a person who dies from malaria frequently dies from lack of blood. Like little, vampires, they eat, they drink our blood. And a lot of people here have probably seen something like this. This is a map of where malaria is in the world in blue, and also where people that have a certain genetic mutation live. And the mutation is for the sickle cell gene. And you notice that there's a large overlap between where people with sickle cell genes live and where malaria occurs, and that's because this genetic change confers resistance to malaria. And this has been hailed, this particular figure, or something like it, occurs in many biology texts, and it's always put forward as a prime example of Darwinian evolution. And it is a prime example of Darwinian evolution. But to understand why it shows tiny changes, kind of like scratches on a car, tiny changes in a pre-existing system, and why it in fact shows the limits of Darwinism much more than its possibilities, I'm going to, I'm afraid, give you a little bit of details about what's going on exactly at the molecular level in this example of Darwinian evolution. And I emphasize in the book that you really have to, if you're going to really rigorously evaluate Darwin's theory, you have to look at where it's acting. And much of the action, although we can't see it, much of the action takes place in the DNA and proteins at the molecular level of life. So inside your red blood cells is hemoglobin. That's a molecule, a protein that transports oxygen from your lungs to your fingers and toes and everything else. And this is just kind of a schematic drawing of hemoglobin showing these sausage-like things, which are things called, which are chains of amino acids together. And that's one representation. Here's a second, I'm sorry. Just for scale, you remember that magnificent complex cilium that I showed you before in the Journal of Nanotechnology. This whole hemoglobin molecule would be one of the blue dots in that structure. So although this looks complex here, it is dwarfed by the complexity of other things. And this is essentially just one, a nut or a bolt in a larger system. Now we can draw the hemoglobin molecule in another form, and I don't mean to tax you again, but I think this is the best way to get the idea across. This is just kind of a writing down the one-letter abbreviations of the amino acids in the chains of hemoglobin. There are 140 in 2 different chains of hemoglobin. In the sickle cell gene, the mutation that gives resistance to malaria, that allows thousands and thousands of people to live who otherwise would die from malaria, one thing has changed. The amino acid at position number 6 right here has been changed from an E to a V. And that's it, okay? Just that tiny, tiny change. Remember, this is just one tiny part, and the cilium contains hundreds of molecules like hemoglobin. This is only one part of a tiny part of a protein. Okay, so this one change allows the hemoglobin to confer resistance to malaria. How does that do that? It turns out that when the E is changed to a V, it's like putting a wad of chewing gum on part of the hemoglobin. Makes it sticky. That allows it to stick to another molecule of hemoglobin in the red blood cell. Turns out there are hundreds of millions of copies of the hemoglobin in the red blood cell, so they all stick to each other and they form a solid. They essentially precipitate. It's kind of like boiling Jell-O, and when you cool it down, it congeals. Well, here the hemoglobin is congealing within the red blood cell. And here's an electron micrograph, a powerful microscopic picture. You see these sickle cell fibers. They're forming inside a red blood cell, where normally it's a nice liquid solution. Now it's like a bag of cement. It's congealed. Here's a microscope picture of normal cells. Here are sickled cells. You see the cell has been severely distorted by this process. Now this is good when a person has one copy of the sickle gene and a malarial parasite goes in a normal red blood cell and it causes it to change shape to the sickle form, because then the spleen of the person recognizes this as distorted, grabs the cell, destroys it along with the malarial parasite inside. Unfortunately, some people inherit 2 copies of the sickle cell gene, one from their mom, one from their dad. And it turns out that when you have 2 copies of the sickle cell gene, you don't need a malarial parasite entering your red blood cell to push it into the sickle form. It will go into the sickle form every time the red blood cell dumps off its oxygen in the periphery and enters your veins. And that can cause the cells to get stuck in the capillary, jam things up, and cause severe problems. And sickle cell disease kills many, many people worldwide each year. So the point is that this paradigmatic example of Darwinian evolution is a tiny change in a pre-existing part of a system which causes severe collateral damage. Well, okay, that's one example. But there are many different types of DNA mutations. The one that changed in sickle cell disease was called a substitution, where you switch that E for a V, one kind of nucleotide for another. It's actually an amino acid in the protein. But you can have mutations that are deletions, where you omit some nucleotides or other things, insertions, inversions, and many other types. And it turns out that some of them have cropped up in the human genome in places that also confer some resistance to malaria. And here's a table showing by far the most prominent ones. Here's the sickle cell mutation that leads to sickle cell disease. Another mutation that confers some resistance to malaria is called alpha thalassemia, in which one gene for one of the chains of hemoglobin is deleted, thrown out. And that leads to anemia, and of course it breaks the gene. But nonetheless, for complicated reasons I'm not going to go into, that allows a cell to be a little bit resistant to malaria. You can do the same thing with the beta chain of hemoglobin. Throw it out, and that helps a little bit against malaria. You can break some other genes, something called glucose 6-phosphate dehydrogenase, G6PD. If that is deleted, it turns out that helps in the fight against malaria. We're not building anything here, though. We're breaking things. We're throwing things out. Another one, BAN3 protein, Duffy antigen. Again, throwing them out or turning them off helps in the fight against malaria. So here's the picture I want to leave with you of Darwinian evolution at work. And this is another picture I grabbed off the web. This is a picture of soldiers, I think it's in Colombia in South America. And it turns out the Army had gotten wind of where a camp was, which was being used by folks involved in the drug trade. And they were on their way to raid the camp, and the folks in the drug trade got wind of it, and they blew up the bridge. Now that's a beneficial change if you are the folks involved in the drug trade. It stops them from invading your camp. Turns out, like many of the mutations involved in malaria resistance are exactly like that at the molecular level. The malarial parasite has to grab onto machinery of the red blood cell to work its way in. One way to stop the malarial parasite is to blow up that molecular bridge, throw it out, and that's what human cells do. But again, we are not making machinery, we're destroying it. Blowing up a bridge might be useful in some circumstances, but it does not tell you how you build a bridge. And this is just to make the point, this is a quote from a paper on malaria and human genetic changes in regard to it, that what I've been telling you about are the best examples of natural selection that we know about in humans. This is what we best know that Darwinian evolution does. So again, much Darwinian evolution proceeds by breaking old genes. Another important point that I'll hit on here is that very few of possible accessible mutations help. There was a handful of mutations on that slide, but there are trillions of changes one could make to the human genome by deleting or substituting or inserting different amino acids or nucleotides. So very few changes help, and the ones that do degrade the human genome. Okay, well, that's one example. But do we have any other examples? And the answer is yes. And one of the best is outside the example of malaria. I'll get back to that. But it's based on work done by a man named Richard Lenski at Michigan State, who's done some excellent work on bacteria in the past decade and a half. And he's just been elected to the National Academy of Sciences for this work. The problem with humans is that they breed too slowly, 10 years, 20 years generation time. But bacteria, like E. coli, Escherichia coli, which lives in guts, can be cultured in the laboratory, and you can grow it with a generation time of half a dozen generations a day. And you can grow it in enormous numbers. And Richard Lenski has done exactly that. And he grew E. coli with a total population perhaps 100 times the number of humans that have ever lived that have had malaria. And he's gone through about 100 times the number of generations, 30,000 generations, then have experienced malaria in recent years in humans. So he has gone from orders of magnitude more generations with a whole lot more organisms, simply to see what evolution would do with these bacteria. And it turns out it does pretty much the same thing as we saw in the case of malaria in humans. In recent years, he's published his findings that genes for something called something that makes a substance called ribose were deleted, destroyed, thrown out by the E. coli genome. Mutations in something, where is it? And another gene that makes a sugar called maltose, they were also thrown out or destroyed. He's recently published work showing a number of other genes, and you see all these little arrows. That's where mutations occur repeatedly in the same gene in different kind of flasks, laboratory flasks that he's growing, showing that breaking these different genes has been beneficial to the organism. So without going into much detail, the general results he gets with many more organisms and much longer generations is what we see in sickle cell. So we are not here building models. We're not hypothesizing what Darwinian evolution should do. This is an observation of what it does do. And what we see, what people have seen before, is that of those mutations that affect an organism, about 99% are detrimental. And Darwinists have always said, yeah, but the 1% that are beneficial, they're the ones that are most important. The ones that are detrimental, you never hear from again. But what we see from sickle cell and the E. coli work is that even the beneficial mutations, the great majority, all of the ones that we see, break genes or degrade function. And here's another little cartoon that I would like to leave to impress in your head. This is what random mutation does. It's a bull in a china shop. The genome, many of the proteins, the molecular machinery in cells is very finely tuned, and random mutation is unlikely to help it. And again, this is not an argument, it's an observation. So let's get back to malaria. And here's the mosquito, Anopheles gambiae, which transmits the worst form of malaria in Africa. And here's an electron micrograph of malarial parasites, the single-celled malarial parasites eating red blood cells and bursting out from them. The nice thing about malaria, at least from one point of view, is that, I said that the E. coli work had orders of magnitude, hundreds of times more organisms than were involved in the human studies. Well, the malarial parasite has even greater numbers. Many more numbers of parasites exist in the world than have existed in, say, Richard Lenski's experiments. I don't know if you all remember your exponential notation, your scientific notation, but here's a little table to show that you can represent big numbers just by putting a 10 and a superscript beside it. That shows that numbers increase very rapidly. For example, 10 to the 6th is a million, 10 to the 12th is a trillion, and so on. Well, how many malarial parasites are there in the world? Well, it turns out that a person who's sick with malaria, when the malaria reproduces and grows inside him, can have on the order of 10 to the 12th a trillion cells, a trillion malarial cells in one person. And every year, hundreds of millions of people get sick with malaria. So if you work out the math, it turns out that there are on the order of 10 to the 20th malarial cells produced each year in the world. Now, to put this in perspective, 10 to the 20th, if Richard Lenski continued his experiments, he would have to continue his experiments for another billion years to get the same number of cells that occur each year in malaria. So the point is that with a huge number of cells, random mutation has that many more opportunities to come up with some lucky change, some fortunate change in an organism for which natural selection can grab onto and run with it. And it turns out that the malarial parasite has wanted to deal with some drugs that humans have invented in the past 50 years or so and find ways around them. The most effective drug against malaria for a long time, for decades, was something called chloroquine. Here's the molecular structure of it. And it kills the malarial parasite. But unfortunately, after a decade or two of using chloroquine, the malarial parasite has become resistant to it. And again, Darwinists have pointed to this as a good example of what Darwinian evolution can do. And again, they're right. But again, just like sickle cell, it points much more strongly to the limits of random mutation than to its capacities. Only about 4 or 5 years ago, the mutations that conferred resistance to chloroquine in malaria were tracked down after a lot of hard work by a number of laboratories. Here's a story from the journal Science from a few years ago, emphasizing that chloroquine is now really no longer effective because of this acquisition of resistance by malaria. And it turns out that that just like sickle cell disease involved specific changes in particular amino acids in a particular protein, hemoglobin, resistance to chloroquine does too. A protein called PFCRT, which stands for Plasmodium falciparum chloroquine resistance trait, it's just a protein that is changed, had a couple amino acids changed. And different proteins from different parts of the world turned out had anywhere from 4 to 8 changes. But 2 changes occurred in almost all of the resistance strain, here and here. And it turns out that having to change 2 or more than 1 vastly increases the difficulty of random mutation finding that combination. For the same reason that, I don't know if anybody here plays Powerball, I'm sure you're all upstanding citizens don't do that. I do it all the time. As you know, if you have to match 1 number, it's easy. If you have to match 2, it goes up exponentially. You have to match 3, it goes up exponentially with the number that you have to match. The same way with changes in DNA. If you have to match more than 1, matching 1 is really not a problem for Darwinian processes. Matching 2, it starts really breathing hard. It has a tough time. For example, here's the frequency of development of resistance to 2 different antibiotics by the malarial parasite, P. falciparum. There is one antibiotic called atovaquone. And it turns out that in a clinical study, if you give it to 3 patients, 1 of those patients will have spontaneous resistance due to a malarial parasite that had a mutation in it, because you only have to change 1 amino acid. So resistance pops up in about every 10 to the 12th cells. But if you have to change 2, you have to get them both right. Resistance to chloroquine arises about in every billionth patient, about every 1 in 10 to the 20th cells. Now it turns out that malaria has 10 to the 20th cells occur every year, so it can overcome this without too much trouble. It's breathing hard anyway, but for larger species like humans or elephants or fish, or where the populations are much lower than 10 to the 20th, it would take many, many, many years to develop a mutation of this complexity. Now this number 10 to the 20th is actually kind of interesting if you know how to do exponents. It turns out that there have been, in the history of life on Earth, the past 4 billion years or so, there have been about 10 to the 40th cells. 10 to the 40th. You'll notice that 10 to the 40th, the exponent is about twice that. So there's good reason to think if you needed, in the history of life, a mutation that was just twice as complex as the mutation that malaria needed to overcome chloroquine, that would be beyond the capacity of life on Earth to generate by random mutation. And again, I'd like to emphasize that what I'm doing here is I'm not arguing that Darwinism cannot make complex functional systems. The data on malaria and the other examples are an observation that it does not. And in science, observation beats theory all the time. So Professor Dawkins can speculate about what he thinks Darwinian processes could do, but in nature, Darwinian processes have not been shown to do anything in particular. Well, I'm going to skip a slide or two because I'm a professor, so I could talk for a long time. And I'm going to say that is where the data stops. Now I'm going to make an argument, and this is the argument. When two proteins have to bind together, like several chains of hemoglobin or the many different proteins in cilia, why do they bind together? The reason they bind together is because their physical surfaces are complementary to each other. These are supposed to be two proteins that have complementary surfaces, and the chemical nature of the contacting groups has to be complementary as well. And it turns out that typically you need a number of different, 5 or 6 different contacts on the protein surface in order to get two proteins to bind together. And each of the proteins, some of the proteins are repeated many times, but each of the different kinds of proteins in a machine like the cilium, they all have to have complementary surfaces that cause them to stick together. I'll skip this one here. Additionally, they don't just have to stick together, they have to assemble themselves. In our everyday world, machines are assembled by people or by machines that have been programmed to do so. In the cell, machines have to assemble themselves. And this is a little essay from the journal Nature of 2 years ago, saying that the ability of cilia and other machinery to spontaneously assemble themselves in the cell, it's as though cars could be manufactured by merely tumbling their parts onto the factory floor. This is not a negligible part of biology. The ability of proteins to stick to each other specifically and with enough force is crucial to their properties. And here's just my little foray into philosophy. Thomas Aquinas was quoted by Cardinal Christoph Schönborn a few months ago in the journal First Things. And Thomas Aquinas compares biology to shipbuilding and says, well, it's the same thing. Nature is just technology, but that itself assembles. And so we can tell essentially that there's purpose behind this because the parts of life assemble themselves. Well, Thomas Aquinas didn't know anything about the cilium, but the more and more deeply we go into biology, the more compelling this self-assembly needs to become. So the argument I make in my book, which I'll just state here, is that the ability to get two protein-protein binding sites is beyond what we would expect from random processes. And yet, in a typical cell, there are on the order of 10,000 protein binding sites. And if you look at the data, and this is data from those studies I talked about, there was only one protein-protein binding site developed by random mutation. That's the sickle cell site. Even in 10 to the 20th malaria, we did not get such a thing. So I argue that the limits of Darwinism, the search for the limits of Darwinism the limits of Darwinism are very strict. That is, that Darwinism explains much, much less of life than I would have thought a decade ago. And on the other hand, I argue that design extends much, much deeper into life than I would have thought then too. Okay, let's just skip over that. Let's skip over this too. Okay, well, proteins, who cares about that? Just biochemists. But most people, when they think about evolution, think about giraffes and elephants and dinosaurs and so on. Large animals. And here's a relatively large animal. This is the fruit fly Drosophila. And a lot of work has been done in the past decade or decade and a half onto how complex animal bodies like this arise from the interaction of proteins and genes. And just skipping over it briefly, the answer is it isn't easy. Just with everything else, the more and more we know about it, the more and more complex it turns out to be. All these names here are different protein parts. And this, almost like electrical or computer diagram, is the interactions that are necessary to build one particular tissue in one small animal. So because of the need for multiple steps to set this up and the coherence of the system, I say that there are good empirical reasons to think that design extends at least to the level of vertebrate class, that is fish, birds, and so on. I would bet that it extends much deeper, but that's as far as our evidence takes us so far. Okay, so that's what I say. But I was really surprised and flattered to find that almost concurrently with my book, a number of reviews of it have been published, some by major scientific journals. For example, the journal Science published a review just last week, a two-page review by a very prominent scientist by the name of Sean Carroll, who's an evolutionary biologist. And we shall say he did not like the conclusions of the book. Well, let me just review very briefly his chief reasons for not liking the book. First of all, he says, "My chief error is that I minimize the power of natural selection to act cumulatively." That is to say that one change is good, next change will make it better, and next better yet. Furthermore, that there are examples of this cumulative selection, including something called pyrimethamine resistance, which is a drug used against malaria. And I omit this, and a notable omission, so kind of insinuating that I'm avoiding data that doesn't fit with the thesis. Well, I'm confused about this because on page 75 of my book, I talk specifically about pyrimethamine. And I say specifically that the first mutation grants some resistance, adding more mutations and can increase the level of resistance. I talk about exactly what he says I'm not talking about. So I'm just confused. He must have something else in mind that he didn't put down on paper. Additionally, I talk about other examples of cumulative mutation. I talk about a hemoglobin called hemoglobin C. Harlem, and say that developed exactly the way Darwin envisioned, by numerous successive slight modifications. And I also say in the book that random mutation is the perfect tool for the evolutionary job when steps are continuous and close together. That is, when you can accumulate mutations. But then I go on to say that, well, if they're not, then it's a problem. And I go on to argue that that is, in fact, the case. So I don't know what Professor Carroll's thinking of. I do speak about exactly what he wanted spoken about. And then he goes on to say that my further argument about protein-protein binding sites, he does not think holds water. As a matter of fact, he says that what I argue is beyond the limits of Darwinian evolution actually is within its demonstrated powers. It's demonstrated. We already know that it can do those things. And he reiterates, it has been demonstrated that new protein interactions and protein networks can evolve fairly rapidly and are thus within the limits of evolution. But if you actually go and read those papers, it turns out they are not demonstrations. They are comparisons of protein sequences between different phyla. They have different sequences, they have different interactions, and the authors ascribe them to Darwinian evolution. And that's the problem not only with the authors there, but with Professor Carroll and many Darwinists. They read into the data their theory. They say, this happens in this organism, this happens in that organism, therefore it must have happened by Darwinian processes. But when we were asking about demonstrations, this is a demonstration. This shows that in an enormous number of opportunities, no such protein-protein binding sites did occur, and that the production of them is a whole lot more difficult than I think Professor Carroll is recognizing. Okay, let me say that this is the end of the biology. In the final chapter, I go off the deep end, if I haven't already, and I talk about kind of more philosophical matters. And I especially talk about how the results in biology fit like a hand in a glove results from other scientific disciplines such as physics and cosmology, which point to the fine-tuning or give evidence of design of the universe for life. And this is supposed to be a picture of the Big Bang, which we now know has to be very finely tuned to allow for a universe to produce life. And it's been called the Goldilocks universe. Can't be too hot, can't be too cold, has to be just right. And this is a very lively topic. Just last year in the journal Nature, Our Universe Outrageous Fortune was an article talking just exactly about that, about how the universe is balanced on a knife edge to allow life to occur. Furthermore, if you read in the philosophy literature, which I try not to do, you see, I hope there are no philosophers here, that in fact the fine-tuning of the universe, two possible explanations are usually envisioned: the design hypothesis and essentially the multi-universe, infinite universe hypothesis. And I talk about that in my book, but let's look at the design hypothesis for a second. The fine-tuning, what we have discovered as science has learned more and more about the universe, and just like the cell in the 19th century, the universe was thought to be pretty bland too, pretty simple. But now we know there are finely tuned laws and constants. But more than that, we know that there are finely tuned properties of chemicals. If water wasn't just what it is, we'd be in big trouble. If carbon wasn't what it was, we would be in trouble. Furthermore, there are what I call finely tuned details and events. And let me kind of skip. These are books that talk about exactly those things, recent books. Let's just look at the finely tuned origin of the moon. These days, astronomers think that the moon arose by something looks like a serendipitous process. Some Mars-sized body hit the nascent Earth. This is supposed to be the developing Earth. Notice that the arrow isn't pointing right to the middle. It had to hit at a certain angle. If it didn't, it wouldn't work. And that was just the right angle to produce our moon. Turns out without our moon, life on Earth would be history for various reasons. And as astronomers, or as Peter Ward and Dan Brownlee discuss in their book Rare Earth, which argues that our Earth is probably pretty close to the only one in the universe that could support life, the getting of that moon to from this collision is a very finely tuned event. My point in the last chapter is that many people think that the laws of the universe were finely tuned to permit life, but kind of want to leave it at that. But if you leave it at that, there's a good chance that that asteroid, or whatever it was, might miss. What happens if you have a great gravitational constant? You have a great charge on an electron, just right, but the asteroid misses the Earth, and now you have essentially a barren planet where the Earth should have been. So in order to produce life on Earth, you not only need the right laws of physics, the right properties of chemicals, you need the right events to occur too. And I make the same argument for the origin of life. The origin of life continues to be unexplained and a real stumper. It's hard even to find a good hypothesis for the origin of life. And the astronomer or physicist Paul Davies wrote in a book about 7 years ago that the problems are so great that it demands some radically new ideas. And my idea, radical and new, or something, is that we can envision the origin of life as just another finely tuned event, kind of like the production of the moon from that collision. And we can envision the changes that occur in biology as more and more and more and more finely tuned events. I know this will strike a lot of people as strange. This is not often an idea you hear, and I develop it in more length in the chapter, but don't have time to go into it here. So here's the picture that I'm kind of trying to paint here in the final chapter. We go from very general laws of nature to more specific, like properties of carbon, to even more specific, like the origin of the Earth and Moon, to into biology, into cells, into molecular machinery, into more specific types of biology, classes, orders, family, genera, and so on. I'm saying that the more that we have known, we knew that the laws, when we discovered the laws of nature, we saw that they were fine-tuned, but the more we knew, we saw that the Big Bang had to have the right speed and right amount of matter. And the more we knew, we saw that certain chemicals had to have the proper they did, and that our planet had to be in the right place in the solar system, and our solar system the right place in the galaxy, and that the origin of life had to be accounted for, and the properties of certain biochemicals. And with Darwin's black box, I essentially say design goes to this depth on the scale. And with the edge of evolution, I say it's much closer, much further down here, that random processes can probably explain changes up to the species level in biology. And somewhere in the middle there lies the broad edge of random evolution. And just briefly, that profound questions remain unanswered, like the problem of evil. Because if you're going to say design extends all this far, you have to admit that some things that don't like us and we don't like them, like malaria, required design as well. So, I'll leave that to theologians and philosophers to deal with. But nonetheless, this problem is not new, and the English poet William Blake, in his well-known poem The Tiger, or at least the first couple lines are well-known, contemplated this too. He's thinking about this magnificent animal with these very sharp teeth and claws. And he says, "Did he who made the lamb make thee? Did he who made the lamb make malaria or other things that can cause us harm?" And my own feeling, for what it's worth, and again, I have a PhD, but I'm no philosopher, is that if you're going to make life, if you're going to set up a stage where intelligent creatures can live their lives and do interesting things and be moral agents, you have to design a stage which will allow that to occur. You've got to build things. You've got to build the platform and so on, and that the platform turns out to be surprisingly intricate and turned out to require a lot more effort than we knew. But nonetheless, that all the world's a stage, that good things, love can happen, and bad things can happen too. So with that overly long discussion, thanks very much for your attention.